let's talk about topic 2.5, membrane permeability. So the structure of our membrane, as we talked before, is selectively permeable. So you have to know that the structure of our membrane is going to result in selective permeability, which essentially means that our cell membrane is separating the inside from our outside. Okay, so it's separating our internal environment from the external environment. So as we come back to our essential knowledge, that's what we have on there, that your cell membrane separates internal from the external. Now, our plasma membrane is acting as a barrier, and you also have to understand why the plasma membrane is selectively permeable, and that it's the structure and how the plasma membrane is formed that makes it selectively permeable. So remember that our plasma membrane, we have our phospholipid heads and our fatty acid tails. Now our heads are hydrophilic, whereas your tails are hydrophobic. The best way to have that bilayer is to have our tails face inside. And throughout our membrane, we are going to have proteins embedded in our membrane. Any substance that can't move through the membrane is going to be able to use the proteins to move across if it needs to. And so we have our channel proteins also embedded within our plasma membrane, which will help to channel substances through that cannot make it between our phospholipid bilayer. Okay, other than our channel proteins, we also have carrier proteins that are going to be embedded, which will take the target cells and basically carry those target cells across. Now, this is the structure. So because of the way that the membrane is built and the way that our proteins are embedded and the way that your hydrophilic heads and the hydrophobic tails are laid out, this is why we have selective permeability. Okay, this is why your membrane is selectively permeable. So what can move across the membrane freely? So we know that the inside is hydrophobic. That means that our hydrophobic, so small hydrophobic molecules, that would mean that they are also non-polar, are going to be able to move through without any additional help. Now, another component though that you do need to understand is that there are some small polar molecules that are hydrophilic that are able to move through the membrane. However, it is in small amounts. Now, this is also essential to life. You don't want a lot of water to make it through the membrane with it, so this also helps to keep the cells from completely losing the fluid. What can't move across our membrane? So hydrophilic substances do not want to pass through the hydrophobic area. Hydrophilic are our polar molecules. So polar molecules, or your hydrophilic substances that are large, are going to need to go through either your channel proteins or your carrier proteins. So your channel proteins are essentially just your hydrophilic tunnels within the membrane that are going to allow specific target molecules through. Okay, so now that doesn't mean that any molecule that is hydrophilic can go through these tunnels. These are specialized tunnels that allow very specific target molecules to go through. So for example, our membrane will have tunnels for sodium ions, and there can also be tunnels for potassium ions. Okay, so these are very specific channels. Another way is our carrier proteins for our hydrophilic substances to go through. The difference between these is that these carrier proteins are going to change their shape in order to move the target molecules and kind of move them through the membrane from one side to another. Okay, so this comes back to our essential knowledge. You have to know that the selective permeability is directly related to the structure. Our small nonpolar molecules like N2, O2, and CO2 will move. Our hydrophilic substances need to have our embedded channel and transport proteins in order to get them to move across our membrane. And you also need to know about our polar charged molecules that include H2O. In order for them to move across the membrane, it does need to be in very small amounts. The last component we need for membrane permeability actually is going to refer to the cell wall. So you should know that the cell wall is going to be the structure that is found only in our plant cells. Okay, so this part of our 2.5 is just referring to the cell wall. You need to understand that your cell wall is made up of complex carbohydrates, where in your plants that's going to be from cellulose, and it's from chitin, which both of them are polysaccharides. The cell wall is going to serve two purposes. One, it's serving as the structural boundary. It is helping to protect and maintain the shape of the cell. It keeps the cells from rupturing if the water inside of the cell, or the internal water pressure, becomes too high, and it helps plants stand up against the force of gravity. Our cell wall also serves as a permeable barrier, so this is kind of similar to when we're talking about our plasma membrane and how the plasma membrane is selectively permeable, so it's not just letting anything into the cell or letting anything out of the cell. In plant cells, in the cell wall, we have something called plasmodesmata, and these plasmodesmata are small holes between our plant cells that are allowing the transfer of nutrients across the cell wall. Okay, so these are nutrients, so it's allowing the transfer of nutrients, waste, or ions through the cell wall. So if we come back to our essential knowledge, you need to know that cell walls are providing a structural boundary as well as being a permeability barrier for substances to the internal environment. You need to know that the cell wall of plants, prokaryotes, and fungi are composed of complex carbohydrates.